Morning, thank you. I'm grateful for the opportunity to talk to you about Capella Imaging and some of the things we're doing around FibroScint. So I'd like to start with what is the problem we're trying to solve. We heard quite a bit about this earlier this morning. About 18% of deaths worldwide are attributed to heart disease. That number jumps to 20% in the US. As a consequence, there are about 90,000 patients a year that are on the list for a heart transplant, and that number is growing every year. Conversely, there's about 2,500 donor hearts available, and that number has been stagnant for a long time, so there's a disconnect there. So left ventricular assist devices, or LVADs, have been used for quite some time now. These are essentially pumps that are used for bridge to transplant to essentially keep you alive until a donor heart becomes available. But as this figure shows, the numbers aren't in your favor. You're not going to get a donor heart. So more and more, they're being used for destination therapy, meaning you're going to live with this. Now, all these devices, the technology is getting better, the batteries are getting better, but they all suffer from a major complication of thrombus, blood clots. And that can lead to one of the most serious side effects is stroke. To counter that, patients are treated with heavy anticoagulants, which leads to another problem, which is gastrointestinal bleeding. So about 30% of these patients are going to require hospitalization at least once a year to be treated for gastrointestinal bleeding. At present, there is no way to assess the level of clot buildup in these devices or to have an endpoint for assessing the effectiveness of anticoagulation therapy. And this is where we think we can step in and fix an unmet need. So this is an image of FibroScint. It's a cartoon where I think I'm the only non-device guy so far. It's basically a tetramer. Each of the arms is composed of a polyethylene glycol polymer terminated with two peptides, one of which binds the radioactive technetium, the other of which binds very specifically and avidly to fibrin. What you see on the right, the rotating image, is a SPECT scan of an LVAD pump that was removed from a human patient that came in for pump exchange. And this is important, that it was removed from a patient. And the bright spot you see at the bottom is a large buildup of thrombus that this patient had, and he was at risk for a piece of that breaking away and causing a significant stroke. So some key concepts I want to stress here. The pumps are all taken from human patients. So this is our preclinical model. We take pumps out of patients that come into the Washington University Medical School for exchange. We implant them into 70-kilogram calves, which are a good model for humans in terms of mass and blood volume, and we acquire images from these guys. So this is as close as you can get to actual clinical data in a preclinical model. The second thing is, as a nuclear imaging agent, the amounts of material we dose are exquisitely small, so there's very little risk of a toxicology concern. So these are the two areas where drugs typically fail transitioning from preclinical to clinical, is either efficacy or toxicology. And FibroScint is very highly de-risked in both of these regards. So in terms of a market opportunity, taking this 90,000 patients a year of looking at heart failure, if we assume 20% LVAD placement, and these are numbers from recent market reports, that market is growing at an estimation of 18% a year, but we fairly modest reimbursement, $1,500. This is a new drug for an unmet need, so that's probably quite modest. We're looking at about $78 million annual revenue first year, expecting that to grow to about 155 years down the road. However, this 90,000 patients, even though it's growing, still qualifies us as an orphan drug. And I don't want to go through all the points on this slide, but there are some advantages to that. And what I think is the most valuable is the agency works with you to get into clinical trials and get first into human. Once you're in humans, it's fairly easy to pivot to follow-on indications, and we think there are other markets that we can be a significant player in this, and those are pulmonary embolism and acute coronary syndrome. So these are not unmet needs, but they're needs that require better tools. And just a couple of figures of merit here. Like I said, these are much bigger markets. The tools that are currently used are essentially designed to eliminate other potential sources of chest pain, and what's left is potentially a pulmonary embolism. So our competitors in this space, for the LVAD indication, there's nothing. It's a completely unmet need. We're aware of another startup in the Boston area that was working on something similar a few years ago. They were using PET imaging, we're using SPECT, and that's outside the realm of this talk, but we feel our SPECT imaging approach is a much better, much more universal in terms of availability of cameras and utility in hospitals. For PE and ACS, competition is the current standard of care, and as I said, there are tools that are used in terms of normal imaging, but they could be done better. So we did a Series A. North Star Medical Radioisotopes invested $1 million in us, and that was used to essentially drive all of our IND-enabling activities. We've contracted with a GMP manufacturing facility and worked out a scalable process to make the drug. We have done a GLP-compliant tox study and gotten clean results. We've done design of experiments to optimize the radio labeling. We're all ready to go. We just need to fund what we're doing, and that's why we're here. We're looking to do a $10 million Series B equity round, which will drive us through beginning our phase 1 clinical trials. Simultaneously, we will be manufacturing GMP material to do a phase 2 clinical trial. Our key opinion leaders in this area have suggested that it's likely we will be able to exit at the end of phase 2, that we would not be required to do a phase 3 study. Again, with a nuclear imaging agent, the numbers are a bit different. The requirements for getting to humans are a bit different. You only need a single species of tox, and that's because, again, this mass that's dosed is so small. North Star Medical Radioisotopes has already expressed interest in this for acquisition, but we're talking with other people as well. A little bit of time on the team. Capella, which is myself and 3 of the other founders here, we're very deep in drug development from discovery to commercial, both small and large pharma. Greg Lanza is a cardiologist at the WashU Medical School. This product actually came out of his lab and was fully exclusively licensed to us from the Washington University Medical School. Our scientific and medical advisors really want to stress this. These guys are world class. Barry Siegel has been working with the FDA on nuclear imaging for his entire career and is just a household name there. Sally Schwartz has authored a number of the guidance documents around experimental INDs and nuclear imaging. And Drs. Ewald and Ito are both very well regarded in their fields and are very active in supporting the development of this product. And all these people are behind it, believe in what we're doing, and are willing to talk to people about it. So we feel very fortunate to have them on our side. So with that in mind, if anybody's actually interested in a drug instead of a device, I hope to see you later. Thank you.